are all aware of the pandemic disease COVID-19. So now up to today, there are many cases proceeding there, more than 1,000 cases in India, in all other countries. But how do this happen? Everyone get tested. But what's actually happening inside a laboratory? Why do they get more tests? And today we are going to speak about only that. What is happening inside laboratory? There are many tests, everyone is proving like PCR tests, everything. But today, as an elaborate thing, we are going to see about the PCR test. Still now, the main reason is no medicine found, either antiviral or antibacterial. So let's go. COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus. When a person is infected, the most common symptoms include cough, fever, and shortness of breath. To start the test, samples can be collected by nasopharyngeal swab or oropharyngeal swab. In nasopharyngeal specimen, the swab is inserted into nostril and gently moved forward to nasopharynx. Then it is rotated gently for the specified period of time to extract the secretion of the virus. Once the swabbing is applied, swab is kept immediately on the sterile tube containing the viral transport medium. The standard method of coronavirus testing is polymerase chain reaction (PCR), which is a method that is widely used in molecular biology to make million to billion of copies of a specific DNA fragment rapidly. Coronavirus contain an extraordinarily long single-stranded RNA genome.
To check these viruses with PCR, RNA molecules must be converted into complementary DNA sequences by reverse transcriptase. Then, the newly synthesized DNA can be amplified standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast, and effective isolation.
To extract viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with lysis buffer. This buffer is highly denaturing and is usually consists of phenyl and guanide isothiocyanate. And also RNAs are usually present in lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulverizing and incubated room temperature. Then the virus is highest under the highly denaturing conditions provided by the lysis buffer. Once a sample is highest, a purification procedure is carried out by using a spin column. The sample is loaded and the spin column, then centrifugation is performed. The procedure is a solid phase extraction method in which the stationary phase consists of silica hydrate and optimal salt and pH conditions. RNA molecules bind to silica gel membrane and at the same time proteins and other components are not retained.
After centrifugation, the spin column is placed into a clean centrifuge tube and the filtrate is discarded. Then the wash buffer is added. The column is put into centrifuge again, forcing the wash buffer through the membrane. This removes any other remaining impurities from the membrane and leaving only the RNA to the silica gel. Once the sample is washed, the column is placed into a clean microcentrifuge tube and the elution buffer is added. Then a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column, and a purified viral RNA, which is free of protein, inhibitors and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for the PCR amplification. In the next step, a master mix is used, which is a pre-mixed concentrated solution that consists of buffer, enzymes, reverse transcriptase, nucleotides, forward prime, reverse prime, Tacon probe and DNA polymerase. Finally, to complete this reaction mixture, RNA is added to it and the tube is mixed by pulse vortexing. Then the allowing the analysis of several samples at the same time. Then the PCR plate is placed into the PCR machine, which is essentially a thermocycler. Real-time RT-PCR is used for the detection of new coronavirus disease 2019 by the amplification of target sequences of RT gene, A gene and N gene. The choice of target sequences depends on the primers and probe sequences. The first step in RT-PCR is reverse transcription. The first-time complementary DNA synthesis is primed by the PCR reverse primer, which hybridizes the complementary part of the viral RNA genome. Reverse transcription then adds DNA nucleotides onto the 3' and the primer synthesizing DNA complementary of the viral RNA. The temperature and the duration is dependent upon the primer, the target RNA and the reverse transcriptase used. Next, an initial denaturation is applied, causing the denaturation of the RNA-DNA hybrid. The temperature and the duration is dependent upon the primer, the target RNA and the reverse transcriptase used. Next, an initial denaturation is applied, causing the denaturation of the RNA-DNA hybrid. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase.
PCR consists of a series of thermal cycles, which each cycle consisting of denaturation, annealing and extension.
Denaturation step consisting of heating the reaction chamber to 95 degrees Celsius and is used for the denaturation of the double-stranded DNA template.
In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing an annealing forward premier to its complementary parts of its single-stranded DNA template. The annealing template relies directly on length and composition of the premier. The extension step, DNA polymerase synthesize a new DNA strand complementary to the DNA template strand by adding free nucleus type from the reaction mixture that are complementary to the DNA template from 5' to 3' direction. The temperature at this step is depend upon the DNA polymerase used after the first cycle is the double-stranded DNA target is obtained. Then the denaturation of the double-stranded DNA is performed, yielding to single-stranded DNA molecule. In the next step, the reaction temperature is lowered, allowing annealing to premier to its single-stranded DNA template. An annealing of the tagment probe of its complementary parts of the DNA tagment probe consists of fluorescence attached with 5' and oligonucleotide probe. The fluorescence emitted by the fluorophore when it is excited by the cyclic light source. The probe consists of quencher with 3'. The quencher allows the direction of the fluorescence. The extension step, DNA polymerase synthesizes a new DNA strand. The polymerase reaches the tagment probe. Its end of gene is 5' nucleolus activity, cleaves the tagment probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released and resulting in an increase of fluorescence intensity proportional to the amplificant synthesizer.
This method allows the estimation of the amount of amplificant synthesized. This method allows the human sequence present in the sample. The number of double-stranded DNA pieces is doubled in each cycle. Therefore, PCR can be used to analyze extremely small amount of samples. To the measurement of the fluorescence signals, a tungsten halogen lamp, excited filter, mirror, lens, emission filter,
charged coupled device CCD camera are used. Filtered light from the camera is reflected off mirror, is passes through a condensing lens and is focused at the center of each one. Then
The fluorescence emitted and reflects of the mirror. A charged coupled device CCD camera are used. Filtered light from the lamp reflects of mirror can be detected by the CCD camera, and each cycle of PCR light from fluorescence can be detected by the CCD camera, which converts the light captured into digital data. This is known as real-time PCR, which monitoring the process of the PCR as required in a real time.